Welcome to The Perfect Stool, Understanding and Healing the Gut Microbiome. This is your host, Lindsay Parsons, and today I'll be talking with Sabine Hazen, MD, founder and CEO of the Malibu Specialty Center, Progenibiome, and Ventura Clinical Trials, where she conducts and oversees clinical trials for cutting-edge research on various medical issues. She's board-certified in gastroenterology, hepatology, and internal medicine, and is a top clinical investigator for multiple pharmaceutical companies. She also acts as the series editor of Practical Gastroenterology on the Microbiome, a peer-reviewed journal that reaches 18,000 gastroenterologists, and is the lead author of the 2020 book, Let's Talk Shit, Disease, Digestion, and Fecal Transplants. But before I get started, if you haven't yet followed or subscribed to the show, be sure to do so. And if you want to get transcripts of the podcast, pop over to my website, highdeserthealthcoaching.com, and sign up for my newsletter. You'll also get my free e-booklet, Finding Your Root Cause Through Stool and Organic Acids Testing, when you sign up. And if you haven't yet done my quiz on which stool test would help you get to your root cause, you can find a link in the show notes and take that. Now on to the show. Welcome to the podcast, Dr. Hazen.
Thank you.
Thanks for coming on.
My pleasure. Thanks for having me.
Yeah, my pleasure. So your book, Let's Talk Shit, Disease, Digestion, and Fecal Transplants. I noticed when I was looking at it that one of your co-authors is Thomas Barodi, yes, who has been doing fecal transplants for years in Australia, right?
Yeah, the master and pioneer of fecal transplant.
Yeah. Well, so funny story about him. I've been obsessed with fecal transplants and this whole field for years, well before I was working in it. My previous career was in international education, and I was actually working at Georgetown University in the Center for Australian, New Zealand, and Pacific Studies, and realized that there might be a way, because we used to bring in visiting scholars and people to do talks and such, realized there might be a connection there since he was from Australia, in which I could somehow reach out to him.
Oh, that's funny.
So I did. I like dug through old papers. I found his email and I wrote him and invited him. Well, I asked my boss first, who was interested in the topic because he actually had some GI issues himself. And I invited him to come and give a talk at Georgetown, and he agreed to it in theory. But then I left that job, and I'm not sure. I don't think that ever happened in the end.
Yeah, he's a great guy. He's definitely responsive to anybody calling him. Over the years, I cannot tell you how many gastroenterologists have said to me, oh my God, Dr. Barodi held my hand on my first fecal transplant. I was so scared. Patient had C. diff and the colon looked awful. And I was so scared to just put poop in there. And he just held their hands. And that's what he does. He's been a mentor to so many of us, and he's so open with his ideas and his innovations, and probably too much because people take advantage of him and use his idea to make a business out of it. And, I kind of started collaborating with him, and I said to him, we need to educate the people on what's coming, and we need to educate them on the microbiome. But I don't think, and it happened during the period of COVID in January 2020, where I thought the end of the world was coming, is when I finished our book with Shelley Ellsworth, who basically helped us. And when we finished the book, I said, I'm not going to call it Let's Talk Microbiome. I'm going to just call it what it is, so people are aware, because this is an emergency. And also in the book, there is a chapter that gives you an idea of how to survive COVID. So, and we actually mention in there the microbe that we believe could be protective against COVID. So, and in fact, there's a publication coming from my lab with those microbes, lost microbes of COVID-19, and that microbe is in there. So,
and what is it?
So we started talking. So if you go into, I forgot which chapter it is, but at some point, the first third part of the book, I talk about the importance of bifidobacteria and how people that have obesity tend to have low bifidobacteria. And if you think about it, that's the same population that's been hit with COVID, right? Mm-hmm. And the first bacteria that kind of popped up as being lost in patients with severe COVID was actually bifidobacteria. And
that's also something that decreases with age, right?
Yeah, and also something that decreases with age, is decreased in autoimmune processes. It's a very important microbe that is in our gut. In fact, it's the microbe that sustains the whole billion-dollar industry of probiotics, right? If you look at the back of all these probiotics, it says bifidobacteria. If you look at kefir, it says bifidobacteria. How many products on the market say they have bifidobacteria, right?
Right, right.
And so that was the beginning. So to me, it was so important for people to read the book because I said to myself, this is going to give them an idea about gut health and how to survive COVID. And at a time where we didn't even have vaccinations or any treatment. And so that's why I wanted it to be a catchy name. And also I figured I'm embarking in a world where I'm challenging the narrative a little bit. Dr. Barodi and we're scientists. So we are the rebels that are going to look the other way. When everybody's looking to the right, we'll look to the left for the solution, right? Because if everybody looks to the right, then you never find anything, right? So imagine like you're looking for gold and everybody's looking in the same spot. Well, you're never going to find anything.
Yeah. So how did you get involved with FMT?
It was interesting. So my friend, Neil Stolman, who was my mentor and a couple years older than me, and when I was in resident, he was in fellowship of GI at University of Miami in Jackson Memorial Hospital. And he was kind of like, I looked up to him as a GI doctor and I wanted to be like him when I grow up kind of thing. Mm-hmm. So I went into GI because he mentored me because I was always impressed by his way of being a physician and his vision that nobody's really right about anything, that we need to be looking constantly for solutions, and no science, no research is wrong. Everybody has an opinion. And so when I was a fellow at University of Florida, he took me around the posters. I was presenting my own poster. It was visceral hyperalgesia at the time. And he took me away from my poster and he said, look at this data. The future is in the microbiome. But he didn't say it like that. He said, the future is in shit. And I said, Neil, if you bring me down that path, I'm going to hate you. And basically what happened is he went that path, right? He started speaking. He would always invite me to all these meetings, ACG, and he was always like the main speaker. And I remember, and I would do the clinical trial side that was cleaner, with pharmaceutical companies. I would basically do the new antibiotic for C. diff and the new pill and this and that. And when the pill wasn't working or the clinical trial wasn't working, I would go to fecal transplant. And back then, it was I remember calling him for my first case that I was doing. And because I didn't call, I didn't know Dr. Barodi at the time, and wasn't going to call Australia. So I would call Neil and I would say, Neil, how do I do this? He goes, figure it out. And, I read all the literature and then I figured it out, and I figured out my own little protocol per se. And my first case was a physician, and I was shocked. When you see a colon that's a disaster inside with ulcers and bleeding and mucus, and you go, oh my God, I'm just going to put stools in there and then it's going to improve it. And then sure enough, a week later, a month later, the patient is better. They stopped having diarrhea and something happened, right? And that was the first case for me. But I still didn't like doing it because I had to put Noxzema in my mask. Nobody likes to play with stools.
Yeah.
So I was still the clinical trial girl. And and I would tell my patients, look, I'm going to put you in a clinical trial for C. diff. And if the trial doesn't work, then I'm going to do fecal transplant on you. And I would use the funds from pharma to basically pay for the analysis of the stool donor and everything. In other words, like they would pay me to conduct the trial. And I would use that money to look for perfect donors for these patients and their families, or using the spouse and making sure that the spouse was clean stools, right? And that's how, when clinical trials became fecal material in the capsule, one could say me and Neil kind of joined forces because clinical trials became fecal transplant in a way. Mm-hmm. And so I joined the shit business, and then I said, well, if we're going into the pharma world, we better start looking at these microbes carefully, because what the complication of fecal material in the future, and what are we doing long-term to the patient, short-term, long-term. Certainly I saw cases of personality changes with fecal transplant, BMI changing post fecal transplant, inflammatory changes, post-fecal transplant. And then you start reading from other physicians, Colleen Kelly and Sahil Khanna and Paul Fierstadt, and you start reading all these other improvement or side effects, right? So you start educating yourself and you have all these questions that are not answered. And when those questions were not answered for me, I started wanting to understand the microbiome. Mm-hmm. More personal in a way, because in my family, I had family members that I wanted to understand what was going on in their microbiome. And when I sent their stools to different labs, I wasn't getting the same validated results. And not only that, but even in the bioinformatics pipelines were different. And so I asked the questions, well, how do we know what's working and how do we know what we're doing if the pipelines are not even validated and these stool labs are not even validated? And so I set myself on a mission to understand the microbiome, especially after I had a case of Alzheimer's where the patient remembered his daughter's date of birth after fecal transplant. And I did it for C. diff. I published that paper. It actually took probably about 5 years to get approved and published because they didn't believe that the patient remembered his daughter's date of birth. I actually had to send them the mini mental status of the patient with the square and the triangles that are drawn perfectly fine. He went from a mini mental status of 20, 21 to 26 and then to 29 after 6 months. So to me, that was like one of those N of one that you go, wait, something's happening in the microbiome with Alzheimer's. We got to pay attention.
And then he had C. diff. That was why he
had C. diff. That's okay. How we were able to do it.
Right.
And so presented that case to all my colleagues and they're like, wow, that's an amazing case, but that's an N of one. We've not seen that. Mm-hmm. And of course, you saw the case of Colleen Kelly with alopecia areata in 2 patients. And then next thing you know, the patients grew hair and you had to ask yourself, well, what grows hair in the microbiome space, right? Because Dr. Baroudi tried to do that on another patient and it didn't work. So which makes you wonder, well, maybe donor matters. Maybe the microbes matter that we're implanting. And so that was the importance for me to create Progenobiome. With the interest, and I realized, I was shaking the beehive a little bit, because every time you start something that's new and it's a physician on the front line of clinical research doing it, it is shaking the beehive of what's already there, right? Because if you find answers, then you may not want to, there's a whole industry that's going to be gone, right?
Right.
Especially if, let's say, we find answers for Crohn's disease in the microbiome space. Well, will the biologics disappear from these pharmaceutical companies? Mm. But I always believe, and that's always the fear, and that's why there's always like powers that be that try to destroy the innovations, right?
Right.
But I'm a big believer that the same way that the post office works and email works, we didn't decrease the work of the post office by bringing on emails. We just expedited the mail transfer back and forth between people in writing, right?
And then started mailing everything in packages and ordering everything.
Yeah, but the mailmen are still busy. And, I always joke, I say, we used to think like, well, we create internet, it's going to remove the need for the post office and it's going to remove the need for books and library and papers, right? But the reality is you're busy in the real world and you're busy in the virtual world. My desk is full of papers and my email has over 2,900 emails that I need to deal with. So I've got this stack of papers on my desk that I need to deal with, and I've got these stack of emails I need to deal with. So yeah. I think in general, we've complicated our lives as human beings, period.
Yeah. The pharmaceutical companies will find something else to work on if they don't need the biologics
I believe that
anymore.
We're here to help pharmaceutical companies, right?
Yeah.
We're here to help improve them, improve their outcome. In medicine, it's never a one pill solution. It's never a one treatment solution. You have patients that respond to Remicade, for example, but then they need nutritional supplements. They need psychologists to deal with their trauma that started off the stress level that created potentially the dysbiosis in the gut, right? So It's never a one pill solution. It's never a one treatment. That's why a lot of these patients, they need their psychologist, they need the nutritionist, they need their acupuncturist, they need all the ancillary support to help them function, because it is a complex disease for a lot of people.
Yeah. So let me stop you for a second. I know most of my listeners are struggling with gut health issues, or else you wouldn't be listening to a podcast that is this inside baseball on gut health. So I just wanted to let you know that in addition to being a podcaster, I work one-on-one with clients around gut health issues like IBS, IBD, SIBO, SIFO, or invasive candidiasis, constipation, diarrhea, soft stool, food sensitivities, and other health issues like autoimmune diseases, skin issues, chronic fatigue, and fibromyalgia. I help clients locate the least expensive lab tests that they can order themselves online to determine the root cause of their issues, then educate them on protocols used by practitioners to address those issues. The first step in seeing if health coaching might be right for you is setting up a free 30-minute breakthrough session with me. And you can tell me what you've been going through, and I'll let you know if it sounds like something I think I can help with based on what you've already tried. So you can find a link to set up a free session in the show notes. And just ask, how successful is FMT as opposed to antibiotics for C. diff?
Very successful. So antibiotics, so what we've come to discover at Progenibiome is that if you look at the genetic sequencing of the microbes of individual, you will notice that a majority have non-pathogenic C. diff in their gut as their fingerprint, right? Mm-hmm. So if C. diff is in my, and in fact, it's in my gut, it's in all the GI doctors that have analyzed their stools. So one wonders, well, did we get colonized with C. diff? You know, we're exposed to patients. Did I get C. diff somehow in the GI lab, touching the handle, touching the door knobs, etc.? Is it part of our signature microbiome, right? If it's been part of our signature, and I really believe that it's been part of our signature for thousands of years, I think we all have C. diff in our gut as a baseline microbiome, because if you look at C. diff, it's 10 million years old, at least, right? They found it in one of the studies I remember seeing. So if you've got C. diff in your gut and you're taking an antibiotic, what are you doing with antibiotics? You're basically depleting the other microbes, right? So you're allowing, in a way, C. diff to become pathogenic, to secrete its toxin, right? So because if you look at the microbiome of patients with toxigenic C. diff, you will notice that they have a lower diversity than everybody else, right? Mm-hmm. How did that diversity get killed? Well, it got killed with the antibiotics we gave them. Right. So what am I doing when I give vancomycin? Well, I'm killing the diversity of the microbiome. So if you look at patients with vancomycin, their diversity is much less than a healthy individual, and even lesser than a patient with C. diff, because you just gave them the antibiotic. So what are you doing when you do fecal transplant? You're not killing the diversity. You're replenishing the diversity. So the message here, why is fecal transplant helpful with C. diff, is because we're replenishing the diversity of the human being. We are giving the human being a new garden in their guts. Mm-hmm. We are removing the weeds, which was the toxigenic C. diff that was taking over the gut because we killed everything around it. Imagine it's like basically you've got this group of microbes and you just killed off all its families. Well, what is it going to do? It's going to try to kill the host now, right? Mm-hmm. You just killed off all its families. So now what do you do when you're replenishing? You're replenishing new families. You're calming that little microbe at a simplistic way, right? Mm-hmm. You're appeasing the balance of the microbiome system. And therefore, the individual is healthy again.
And do you use antibiotics prior to doing the fecal transplant?
Yes. So you always want to kill off as much as everything, because you're going to give a new microbiome.
And how long do you use and which one?
So I usually do either vancomycin or fidaxomicin, depending on coverage of the patient. Sometimes I'll do flagyl. It's not really great, mostly because most patients can't tolerate it. But usually vancomycin or fidaxomicin.
For how long?
For 10 days, 10 to 14 days. And then basically I do fecal transplant. Mm-hmm.
And are you finding it hard to find donors who are qualified? Like, will you take a donor that has C. diff?
No.
Right. Presumably not, right?
Wait. You mean non-pathogenic C. diff?
Yeah.
Oh, I'm going to find, yes, all donors have non-pathogenic C. diffs in their gut.
Okay.
In the genetic sequencing. And this is a very, very important thing to mention. Toxigenic C. diff is the C. diff that secretes the toxin and therefore causes diarrhea. Mm-hmm. Non-toxigenic C. diff is just a fingerprint of the microbe that's really doing nothing in your gut. So even if you find it in the gut, doesn't mean that it's doing anything.
Is it a different strain then? What's the name of the strain?
No, it's still the same bacteria, but it's not potent, right?
But how can you discern that in a
Well, in a genetic sequencing world, you have to do a messenger RNA pipeline to basically see if it's reproducing. In a and that's in a research world. In a clinical world, you have to do a PCR to see if you have toxigenic C. diff.
Okay.
So
who does that test?
Oh, anybody does that test. All the GI doctors do that test. So if you have diarrhea and you're basically, you're going to go to your doctor and your doctor is going to do a C. diff by PCR to look for toxigenic C. diff.
Okay. Got it.
And they're looking at that point for a specific strain that stimulates that basically if you've got the diarrhea symptoms and you've got the C. diff positive by PCR, then by all that's C. diff in the patient. Mm-hmm. What we look at is the genetic imprint of C. diff. That doesn't mean that C. diff is doing anything. It's most likely doing nothing, especially if the patient is asymptomatic. When you look at a patient and a donor, you have to do a whole bunch of workup, right? So the first thing is obviously you do a GI panel. You want to make sure you don't have C. diff and that donor, toxin A and B. So you'll test for C. diff toxin A and B. You're going to look for adenovirus, Campylobacter, E. coli, Entamoeba histolytica, E. coli, Salmonella, Shigella, Vibrio cholera, Yersinia. There's a lot of bugs that live in the gut. You want to make sure they're not active in your donor, because if you give those stools to a donor and there's a lot more microbes, obviously, especially now with COVID, you have to make sure the donor doesn't have COVID, right, in the stools. Because if you're giving it to an immunosuppressed patient who has C. diff to begin with because he's immunosuppressed, you could kill him. And that's why we've seen the 4 cases post-fecal transplant that died.
4? I didn't realize there had been 4.
Cases. Yeah,
was that from that same single donor?
No. There were 2 other cases that were brought up probably in the last 2 years, I think.
Oh,
there's been 4 cases altogether.
And were they from? I think the originals were from E. coli, right?
Yes.
And what were the others?
I can't remember what the other 2, but I remember it was like 4 cases.
Okay.
I don't think the other 2 they even knew what it was. They were concerned about, the patients were extremely immunosuppressed to begin with. So in those patients, you have to try, no matter what the risk, to look at risk benefit ratio.
Right.
But definitely that brought up the idea of looking for vancomycin resistant E. coli. And that looked up, and then that's basically why we are doing all these tests. And now with COVID, look, we were looking for a donor recently in one of my patients that had C. diff, and I used her daughter as a donor, and lo and behold, I found COVID in her stools, so, I can't donate it to her elderly mother with COVID in the stools.
Of course.
So, so it is becoming challenging to find good donors. It is also becoming challenging because with COVID, the microbiome changed. With potentially vaccination, maybe the microbiome is changing. With the stress that people underwent, the post-traumatic stress from COVID and the quarantine and the wearing masks, the microbiome changes. Certainly wearing these masks all day long full of infections, is not really helping your microbiome because they're infected and you're just breathing in all these germs that are in the mask. So all that affects your microbiome, in my opinion. And so it is difficult to find good donors right now.
Yeah. No, I've had a few people who've come to me wanting to use a relative for a fecal transplant, just doing it on their own. And, I told them what tests to run, and invariably they show up with H. pylori and with C. diff and with all these other things. And I'm like, I can't recommend that you use that person.
Well, that's it. That's the problem, right? I mean, it's like that daughter that was healthy to begin with, but then I found COVID in her stools, right? So I'm not going to be giving a stool donor with 4,000 copies of COVID that I found in the stools to a little elderly woman, God knows that would shut down fecal transplant really fast. So we have to be careful. So, and it's funny because even the procedure, we joke in the GI lab because sometimes we're sterile. We're very clean in the way we process putting the stools into the colon, right? Mm-hmm. And things happen and you're like, I can't believe I'm that sterile because I'm dealing with poop to begin with. But we have to be sterile because unfortunately the microbiome is a fragile, it's not meant to be put back, right? Mm-hmm. Fecal material is meant to be out. That's why God created us to have colons that evacuated our secretions, right? The bad stuff from our bodies get out, right? So that's the whole process of putting it back into the earth, right? And then the earth processes it, all these microbes, right? It was never meant to be put back. The fact that we're finding some improvement with C. diff, to me, I think that C. diff was really the can that got opened to look at the microbiome and to look at the destruction that we're doing, mm-hmm. to say, hey guys, you're killing the microbiome and C. diff is popping up in these people. And all these bugs, by the way, that are super resistant, these virulent bugs, mm-hmm. bugs don't just become super bugs. Right. Unless we do something to them to make them super bugs. And I think that's the most important thing to reflect on, right? Especially with COVID. COVID that just didn't happen, right? The human microbiome has got to be pretty messed up for COVID to penetrate and create disease to begin with, or create people to die. So when you look at the people that are affected, the autoimmune people, the people with cancer, the people that are elderly, the people that are overweight, there is a picture there that tells you dysbiosis of the microbiome, penetration of COVID. Penetration of COVID wouldn't happen if that dysbiosis didn't happen. Mm-hmm. I really believe that if you have a strong microbiome, and those are the people that are fascinating to me to study, and you've probably seen me on Twitter saying, if you've not been vaccinated and you've not gotten COVID, please call me. I think these are the people that are super fascinating to study because, in my opinion, they hold the mystery to why they survived COVID.
Oh, well, that would be my sister.
Yes, I'd love to test her stools because
okay.
Those are the people that are
I'll send her your way.
Yeah, no, I mean, it's fascinating because I've kind of, I've been the guinea pig on this pandemic. Mm-hmm. And, I test my stools on a regular basis because I own the genetic lab. Mm-hmm. And I have to tell you, it's fascinating to see my microbiome progress over time during the pandemic. I mean, it's been fascinating to watch. So, because I test, if I take a medication or if I get a vaccine or if I get anything, I look at my microbiome. What is my microbiome doing while I took something?
And yeah, what kind of differences do you just see over time?
Well, that's going to be published. So I can't really talk about,
okay.
but you are going to see it because, like I said, I am doing my timeline and I've been following myself and what I've done and what it's done to my gut. But I think also we've been following the guts of a lot of people. And we've also looked at, before the pandemic, we had probably over 1,000 stool samples. And we're going to be looking at after the pandemic to kind of say, okay, well, what is different, right? Because I think that's fascinating data as well. And also correlating it with those patients, whether they got vaccinated, whether they got treatment. Some people are taking hydroxy, some people are taking ivermectin, some people are getting boosted and vaccinated. So it's important to look at it to see what is all that doing to the microbiome and then get a better idea of what the future is going to be and how to survive the next bugs. Because you can imagine if you've gotten COVID, there's obviously a dysbiosis in your gut that predisposed you to having COVID. So now the next virus that comes around has a potential of becoming super virulent in that person. So that's why when you follow people who have gotten COVID the first time, then they get COVID the second time. If we don't address the microbiome dysbiosis, it's a domino effect constant until, you wake up one morning with an autoimmune process or something. And you go, well, what happened? Well, the domino started way back when you had COVID the first time. That was your first sign, right? It's like C. diff patients. You hear of the story of a patient that gets C. diff, and then down the road, they end up and they're given antibiotics. And then down the road, they get an autoimmune process going on. You have to wonder, did C. diff, was that the beginning? Was that the sign that said, you know what? C. diff was my first sign of dysbiosis. I should have paid attention.
But I mean, 80% of Americans at this point have had COVID. So, I mean, how can you say that that's all dysbiosis generated?
Well, is America a dysbiotic country?
Sure.
Is it?
But what percentage of people in other countries? I don't know those numbers. Are they getting much lower infection rates?
Those are the things to look at. I mean, certainly America is high in processed food. They certainly take on a lot of microbes. They're eating tomatoes in December. You look at Europe, not so much. They don't mix microbes so much. You look at the American way of life, high stressful, high go, 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 not as much as, if I look at the Spanish population. And again, Spain is becoming more America today. But if you look at the olden ways where people used to sleep from 2 o'clock to 3 o'clock and rest, and it was like work, family, and pleasure. Now we've become a society that's just go, go, go. We're on our cell phones constantly. We're not doing any yoga, meditation, breathing. We're not outdoor in nature. We're not gardening. Certainly when you look at kids with ear infections and you see kids in a classroom have more diseases and more infections than kids that play in the gardens, right? I mean, we've certainly seen those studies where exposure to microbes from the earth definitely protects these kids, right? Is the American population big on going outdoors, hiking, playing with the earth, gardening? Not really. So 80% of the population, if you look at the statistics of American public, probably the 80% that got COVID is because they were not doing all these things. Yeah. I can tell you that just with the people that I've treated, because I'm correlating that, the people that do amazingly well are my farmers and my gardeners. I have like husband and wife where the wife is gardening constantly, got COVID, mild symptoms, and then the husband's a physician and he's high stress. He barely made it. Mm-hmm. So I think severity of symptoms is also probably correlates to lifestyle and stress factors. Yeah. And definitely, and the stress from the news and listening to the television and, the drama, and the conflicts and all that. What is all that doing to your microbiome?
Okay. Right? Well, let me stop you. Let's dig into some other gut issue stuff. So
yes,
with the testing you've done, have you noticed that there is a pattern of bacterial dysbiosis for certain diseases?
Yes. So, but nothing I can really discuss because it's all preliminary data. So with me, you always hear that because remember, all data needs to be valid, verified, and reproducible. There's definitely a lot of interest. I wouldn't be continuing this if I didn't see something, but we are looking. We're looking aggressively, in diseases like Parkinson's, Alzheimer's, autism, ALS. That's all interesting to me.
Yeah. Okay. Now, on your website, on Progenibiome's website, I see blurb that says, want to learn more about gut refloralization? Contact us below and we'll follow up with you. So I'm wondering, do you have options for FMT for people who don't have C. diff?
We do not. We work with the agencies. We work with the FDA. We submit what's called an IND to the FDA for emergency cases. So, for example, I had a case of metastatic mesothelioma. So I submitted that case to the FDA to allow me to do fecal transplant. They approved it and we did it. So that was one case. We got allowed to do autism in one child. We are trying to get approved to do 30 kids. Mm-hmm. We've been working at it, mind you, for the last 2.5 years. So it's time consuming. It's extremely expensive to put these protocols into the FDA. Mm-hmm. We're lucky because I'm a research center. And so therefore I have a portal with the FDA that I use to submit all those INDs. Mm-hmm. However, it is expensive. It's a lot of back and forth with paperwork. I think, if you look at Alex Karutz, and there's a lot of videos on the Malibu microbiome meeting. There's a lot of videos because I do a lot of lectures with doctors that are doing fecal transplant. There's going to be lectures from Dr. Sahil Khanna, Dr. Barodi is going to be on that website. So I encourage everybody to go to the Malibu Microbiome meeting.
Okay, I'll link to that in the show notes.
Yeah, and this way they can see the videos. Neil Stolman does a great lecture on Microbiome 101 for anybody that doesn't know. Refloralization, I put it in there because it's a vision of what I believe the future to be. Mm-hmm. I didn't like the term fecal transplant. I know it's been retermed. Alex is calling it, Dr. Karutz is calling it microbiome transplant, which is a more appropriate term for it. Mm-hmm. I like refloralization because I believe we come from flora and we go back to flora. You know, the process of dying is our microbes in our gut get stronger and then they decompose our bodies back to the earth. So to me, we go back to the earth. Those microbes go back to the earth. The foods we eat come from the earth. So essentially, we're feeding ourselves constantly with microbes from the earth, so from the earth to the earth. So I believe that in the future, it's probably going to be more of a refloralization procedure in the sense that finding what's out of balance and replenishing it by what's to create the balance. I think that's the pharmacy of the future. That's my vision. So that's why I put the term refloralization. Mm-hmm. We are seeing certain microbes are improved with certain nutrients and certain vitamins and certain products. So that all plays a role in the refloralization process, in my opinion.
Mm-hmm. So assuming, most of the people who are listening to this podcast, they don't have super healthy microbiomes, but assuming they can get back to a state of a healthy gut microbiome, what would you recommend for people staying that way?
Well, I think if you're healthy, then keep doing what you're doing, right? Yeah. If you're healthy, don't do anything because whatever you're doing great, especially if you have a longevity in your family. Mm-hmm. Don't mess that up, right? If you're unhealthy and you have a family history of heart disease, etc., well, that's time to take charge of your health, right? And that means starting to educate yourself, seeking nutritionists, seeking naturopaths, seeking functional medicine doctors, seeking someone that will guide you in a good way to a proper nutrition and also help you with, I like to call it the fermentation of your gut, right? So gut health really is what it's all about. And gut health is not necessarily the same for every culture, right? Because you could see a Japanese is eating Mexican food. He probably won't tolerate it. And a Mexican person eating Mexican food will not tolerate Japanese food. Certainly I've been a gastroenterologist long enough to know that there's certain foods that people don't tolerate. And I think probably we're born with a certain predisposition to eat foods from our culture and our races. And I think that's your comfort food. You should stick to the comfort food. I think anytime people try to change what's working is when they get themselves into problems.
Mm-hmm. Okay. So I've had this theory for a while that issues with vaccinations are often related to antibiotic use and a dysbiotic microbiome. Do you have any insight into that?
No, I stay away from vaccination or discussing vaccination because I want to stay alive. Not that anything would happen to me, but vaccination is a complex product. It's a complex issue. I don't think anybody's really looking at it. I think it needs to be looked at. I think what we learned from antibiotics 25 years ago, antibiotics was given for everything, right? And what we learned from antibiotics is that actually, if you take too many antibiotics, eventually you're going to have a little bug called C. diff because you've killed all your microbiome. You've killed your gut. I think it's definitely established now, and people understand that if you take antibiotics, you have a risk of killing your gut. And that's why the whole probiotic movement came on, because if you're killing it with the antibiotics, you have to promote your gut with the probiotics, right? And thus the movement of the yogurts and the drinks and the probiotics, etc. I think there's going to be a time that we're going to figure out the same thing with chemo drugs. If you kill the tumor, you got to work on the gut to support the killing of the tumor, right? Because you can't just kill. You've got to replenish. And again, that's the same thing we were doing with C. diff. We were trying to kill with antibiotics, but instead we got people worse. What we needed to do was add more microbes. So I think there's going to be a time a time that also people are going to start looking at vaccination and seeing maybe there's something we're doing with vaccination that needs to be supplemented with something else to balance the benefits of the vaccination with this balance that could be happening in the gut, for example. Okay, and I'm not saying that there is,
Right.
but I'm just saying that I think we need to look at it because it's never a one pill solution and it's always domino effect. Action leads to a reaction with everything.
Yeah, and I'll just add to that though that I'm a supporter of vaccination and have had my children vaccinated and myself and all that.
Yeah,
but I'm just I just look at
I vaccinated my kids. I got vaccinated
because there's so many people now because of the politicization of the COVID vaccine now that are anti-vaxxers, and I feel like it's an important point to not completely deny that there's ever been any problem with any vaccination because that's what promotes conspiracy theories and
and I'm not a big conspiracy theorist. And, it's so funny because on Twitter, people think I'm an anti-vaxxer because I ask questions. Because, listen, I'm a scientist. So if I'm going to have a person that comes to me and thinks they have a side effect to a vaccine, it's my job to look, is that what's going on there, right?
Yeah.
So because it's called, in my world of clinical trials, when you have any investigative product, and a patient has a side effect, any side effect, be it, pain in the mouth, be it headache, you have to document that. It's called an adverse event. And if they end up in the hospital or they're having something that's serious, like they're paraplegic, I look at the case of Maddie de Gabe, which, I got involved in looking at her. You have to pay attention to that because that's a serious adverse event. And serious adverse events, we cannot ignore them because they tell us something else in the future. Mm-hmm. So I think it's our job to pay attention to everything. I think also we have to pay attention to the people that it doesn't work for, right? So the people that got vaccinated and still got COVID, we got to ask the questions.
Yeah.
So just because I'm asking the questions doesn't mean I'm anti-vaxxers. It just means that I'm a good scientist for asking the questions.
Yeah. No, I and my children were vaccinated and boosted and still got COVID, but it was Omicron, so that was sort of expected since that was less sensitive to the vaccines.
Yes.
Okay, so you mentioned that the people who were most susceptible to COVID had low levels of bifido, and I'm just wondering because actually I did with Thorne, one of their metagenomic sequencing of the microbiome, and they said I was low in bifido, and to take rice, well, they said, I can't remember the name of the actual fiber, but it was essentially rice bran.
Right.
So that's what I'm taking that. But other than taking probiotics with bifido and that, what else can boost bifido?
So I have to warn on that because I never put myself in the position to kind of say, yes, we found this. It's research, right? So it's my hypothesis. It's a finding. It's a small study. It needs to be done as a bigger, larger scale, obviously. So I don't want people like leaving this and saying, oh my God, I've got to check my bifido to see if I'm right, right? Because until other doctors validate, verify, and reproduce the data, it's still research of one scientist.
Right.
So I think if it doesn't hurt, take whatever was suggested. But if it becomes like a heavier treatment or something that is risky, I probably would stay away from, then not necessarily trust those labs. Remember, these labs are not validated and not clinical, right? They're just a consumer product that's out there, but it's not, the majority of these labs are not even CAP or CLIA certified. So it's very important when you do your stool testing. And remember, we're in the research, we're writing the data, and we're not a commercial stool sample yet because there's so much to learn. So you got to be aware of stool companies that are selling you these tests and that are telling you you have low bifidobacteria because maybe they're trying to sell you a probiotic for one.
Yeah.
I know we have validated a couple of those labs because my patients come to me and say, look, I just took this probiotic from this company and, my bifido was low. And then we compared it and we couldn't find that their bifidos were low. So you got to be careful of anything out there. It's all research. And everybody needs to understand it's all research.
Yeah. Well, it wasn't the first low bifido reading I'd had. I've certainly had other, probably 16S sequencings that showed low bifido as well.
Yeah. So that's good. So you validated yourself.
Right. Over time.
Yes.
So are there any other diseases where you've noticed changes when incidentally you were doing the transplant for C. diff?
We are working on autism right now. We're going to try to bring on a protocol for Parkinson's and Alzheimer's. I'm working with Dr. Sheldon Jordan at UCLA, who is the top interventional neurologist, in my opinion. We're also working with anxiety with Dr. Sasha Bystritsky. So that's going to be an interesting study. We feel that we see something in anxious patients' microbiome. So we're evaluating that closely.
And are you just looking at what is going on in the microbiome, or are you're making interventions?
No, we're looking and we're making interventions.
Through
we're trying.
FMT.
Well,
or not always,
or something else.
Okay.
Yeah. So we're basically, remember, I'm a research center,
Right.
so we have access to animal labs. So right now we're looking at an animal marker for Parkinson's. So we'll see. I mean, the future is exciting. We're seeing some things and we're going after it full blast, and we've been lucky so far. So we're still above the ground as far as finances, but people can support the Microbiome Research Foundation, because that's how we make all these trials possible and this research possible. The paper for COVID, the lost microbes of COVID, or finding COVID in the stools, was paid by the Microbiome Research Foundation. So please put that on the link so that people can support that. Yeah. So that's how we advance science, through research, through donations. We've been fortunate. A lot of patients I've helped over the years that have, I'm in Malibu, so I have an affluent population, and they've been coming forward and been very generous in supporting us. So, and also the patients, the patients support us. So it's been amazing to help people.
That's great.
So, and to see and to kind of go into the research with them and see what we see, and saying, look, it's research. This is what I see. I could be wrong, but I could be right. And this is the beginning, and having that frank, honest discussion with the patients and giving them a consent, because really all our research is consented. Anytime anybody gets tested with us with the microbiome, it's all research. So they have to sign a consent before they get tested. Or if we test their kids with autism, etc., they have to sign a consent. We are supervised by a regulatory board that overlooks all our IRB, that overlooks all our research. We're doing, Howard Young at the NIH said this is the way to do it, and I'm doing it. So I wrote 52 clinical trials on the microbiome and disease. I'm looking at every diseases and every skin condition. I'm very fortunate. My sister is the top dermatologist in New York City. I have another sister who brought Harvoni and ivermectin in the market. So we have a huge database of patients through clinical trials over the years. So we've been able to utilize these databases and put patients into clinical trials to at least get a beginning of a view of what does Parkinson look like in the microbiome, what does Alzheimer's. So we have an idea, and we're continuing slowly, and eventually, as we publish, I encourage everybody to go on the Progenibiome website, because it has publications.
Okay.
We've published about 35 papers, I think, in the last 3 years.
Oh, great.
So, it's moving.
Okay.
I have 52 more papers to go. We have a lot of scientists helping me, and we encourage collaboration from everyone. Anybody that wants to be involved, please, we're all inclusive. Like I said, I'm a research center. I'm a research lab right now, a research genetic sequencing lab. I'm not a commercial. But anybody that wants to get involved and has something that they want to crack the code of, can help us, and together, hopefully, we'll join forces. I'm fortunate. I have a lot of doctors, a doctor from Turkey that I'm working with right now. We're looking at Crohn's and ulcerative colitis together. Dr. Barodi has been an inspiration and definitely a huge mentor and a huge brain and genius.
Yeah. Speaking of him, I would love to get him on the podcast. Any chance you would introduce me?
Oh, yeah, absolutely. I could do a email.
Wonderful.
He's extremely busy right now because he's trying. We started off that controversial triple therapy for COVID with ivermectin, doxycycline, zinc. So, he's busy fighting the wolves himself because he wants to see that going. He was behind the therapy for H. pylori. So patents is his world, and combination therapy is his world. And He's a genius. I'm so fortunate to be working with the man. And, I've done my part, which was like doing the clinical trial with the FDA. So now it's on to him to take it to the next level. Mm-hmm. That was not an easy trial to do, and certainly a lot of
controversy,
controversy behind it.
Right. Okay, well, I appreciate that. Yeah, even if he doesn't have time, maybe he will eventually.
Yes, for sure. Okay. I think, as he kind of like figures out this whole triple therapy, what he wants to do with it, I think that probably would be the best time.
Okay. Well, thank you so much for coming on and sharing about what you're doing at Progenobiome. And
thank you.
I look forward to talking to you in the future sometime.
Yes, absolutely. Thank you so much.
Well, I'll link to Progenobiome and their website, and I just peeked over at some of the publications that were on there, and I see the one she was referring to that Dr. Barodi was involved in is called Effectiveness of Ivermectin-Based Multidrug Therapy in Severely Hypoxic Ambulatory COVID-19 Patients. And then there's a lot of microbiome-related research as well, of course. Anyway, if you would like to connect with me online, you can follow my High Desert Health Facebook page, join my Gut Healing Facebook group, or join my newsletter list at highdeserthealthcoaching.com, as well as find me on Twitter, Instagram, and Pinterest. And links for all those are in the show notes. Thanks for joining me today, and here's wishing you all the perfect stool.